All right, good afternoon, everyone. For those of you who don't know me, my name is Arun, and today I'm going to be presenting on the topic investigating the efficacy of the mycoherbicide Lalcide Chondro in controlling the resprouting of several invasive shrubs and trees here in Southern Ontario's urban and peri-urban landscape. So getting started with some background information, invasive plants are, of course, plant species that are not native to a given area, which cause environmental and/or economic harm. The main environmental impact is that invasive plants are often able to outcompete native plant species, which can, of course, have detrimental impacts on biodiversity and negatively alter the forest ecosystem as a whole. Examples of economic impacts include the high costs involved with trying to manage these species, as well as the impact on the forestry and agricultural industry through a loss of profit. A common example of an invasive plant is buckthorn. Buckthorn is one of the most widespread invasive plants here in Ontario. It outcompetes native plant species by forming dense thickets, among other ways, and it costs Ontario thousands of dollars every year in trying to manage it. It's also important to understand some of the factors that allow invasive plants to spread rapidly. Some of these factors are characteristics of the plants themselves, including things like a high level of seed production, widespread seed dispersal mechanisms, rapid growth, and adaptability to a wide range of environments, as well as even things like allelopathy. Another important factor to consider, and one that is very relevant to the topic at hand, is the impact of urbanization. Urban centers are hotspots of biological invasions, since they are often also transport hubs, which provide a pathway for the introduction of these invasive plants. On top of that, other factors involved with urbanization can also promote the spread of invasive plants once they have been introduced. And this includes factors like a reduced level of competition, a higher proportion of impervious surfaces, high human population size and density, as well as high levels of environmental disturbance. Relatedly, habitat fragmentation as a result of road, trail, and sidewalk construction, and subsequently the edge effect caused by habitat fragmentation, is also, of course, very common in urban environments and is one of the biggest drivers of invasive plant spread. So now I want to introduce the target species in this study. And these species were Norway maple, winged euonymus, tree of heaven, Manitoba maple, autumn olive, multiflora rose, as well as several species of invasive honeysuckles, which have been grouped together for the purpose of this study. All of these species are, of course, invasive, and they are also all either shrubs or trees, or in other words, they are woody plant species. Additionally, like buckthorn, all of these species possess many of the characteristics that allow invasive plants to proliferate. They all are commonly and abundantly found in Ontario's urban environments, and they all have known detrimental impacts on the forest ecosystem. Now, there are a few issues currently involved in trying to manage these invasive species. First and foremost, like many other hardwood plant species, these species have the ability to resprout after mechanical thinning through stump or root shoots, an example of which can be seen in this first picture here. And so given this problematic characteristic, many invasive plant management strategies have adopted the use of chemical herbicides, and specifically glyphosate and tricloper-based herbicides. Now, the issue here is that both of these synthesized chemicals, as many of you know, have known detrimental impacts on the environment. For example, both chemicals can persist for a long time in the environment. They can impact non-target plants and other organisms, and they can also enter waterways, thereby further spreading their negative impact. And so in light of this, biological control agents, referring to the use of organisms to control pest species, have been investigated, and specifically mycoherbicides, referring to herbicides formulated using fungi, have become a major recent interest and is also the focus of investigation here today. So the specific mycoherbicide being investigated is called Lalcide Chondro, which is seen in the first picture here. Lalcide Chondro was formulated by the company Lallemand Plant Care / Bioforest, and it was formulated using the fungus Chondrostereum purpureum, which is seen in the second picture here. This is a fungal plant pathogen that is native to North America, including here in Southern Ontario, and it is, so sorry about this, one second. And it is, sorry, I've completely lost my train of thought. Let me just give me 1 second. Yeah, so it is native here to Ontario and including Southern Ontario, and it causes silverleaf disease in woody plant species. The disease is caused by the fungus blocking xylem vessels and disrupting leaf cells, thereby reducing the water transportability and leaf functioning of the plant. And its main symptom is a silvering of the leaves, as the name suggests, an example of which can be seen in the third picture here. This disease reduces the growth and vigor of the plant and often eventually leads to its death, hence its use as a mycoherbicide. Currently, Lalcide Chondro is mainly used for controlling the resprouting of common and glossy buckthorn, as well as several natives for general forest vegetation management purposes, and it is sold to a variety of groups. Its known efficacy in controlling the resprouting of buckthorn specifically is why this investigation took place, in order to see if these other target species experience similar results when Lalcide Chondro treatment is used. And so this brings me to the objective of the study, which was to see if Lalcide Chondro is effective in controlling the resprouting of these target species. And specifically, this was investigated by looking at 2 response variables, the first being average number of resprouts, and the second, average length of resprouts. So getting into the methods and starting with the study sites, almost all of the sites were located within the Greater Toronto area. These sites were going from left to right on the first map here: Rattray Marsh in Mississauga, High Park in Toronto, David A. Balfour Park in Toronto, as well as the Lower Dawns Parkland, which is also in Toronto. All of these sites are urban parks / urban forests, and so they experience many of the same issues that urban areas in general face when it comes to invasive plant spread. The only site that was located outside of the GTA was Somerville Nurseries, as you can see in the map here. And this was a private tree farm located in Everett, Ontario, which is located just north of the GTA, as you can see. And so it can be classified as a peri-urban environment. All of these study sites contained 1 or more of the target species in a very high abundance. Looking at these pictures here, this first one is of winged euonymus in High Park. The second is of Manitoba maple at Somerville Nurseries. And the third is honeysuckles in Rattray Marsh. And so looking at these example photos, we can see just how abundant these species are within these target sites, and they were often very densely packed as well. To give a further example, when looking at honeysuckles specifically, we found a 5 meter by 5 meter area that contained over 30 individuals. So this exemplifies just how problematic these species are within these study sites, as well as in urban areas in general. The fieldwork component of the investigation was done by setting up either 6 10 meter by 10 meter plots or 12 5 meter by 5 meter plots for each of the target species. Half of the plots were designated as treatment plots, while the other half were designated as control plots. Both plot types involved cutting the target species 2 to 3 inches above the ground, with the treatment plots then having the mycoherbicide paste applied onto the freshly cut stumps of each of the target species, an example of which can be seen in this first picture here. The cutting and treatment occurred in early June, and the first and only post-treatment check looked at in this investigation occurred in late August. So there was an approximately 3-month window between treatment and post-treatment data collection. During this post-treatment check, data was collected on the number of resprouts for Norway maple and winged euonymus, as well as the length of resprouts for all of the target species. Data was then analyzed using ANOVA's and Tukey's post hoc test to determine if significant differences existed between treatment and control for the various species. So getting into the results and starting with the response variable that was looked at for all of the target species, which again was resprout length, we can see from the graph here that average resprout length was lower in the treated winged euonymus, the treated multiflora rose, the treated honeysuckles, and the treated Manitoba maples. However, average resprout length was greater in the treated Norway maples and the treated autumn olive. Lastly, there were no resprouts at all on any of the Tree of Heaven stumps when resprout data was collected. So clearly there is some variance here in the pattern of average resprout length between treatment and control for the various species. But of course, the more important question here is, are any of these differences significant? And so getting into the data analysis, it was found that significant differences in average resprout length between treatment and control only existed for winged euonymus, Manitoba maple, and the invasive honeysuckles. However, all three showed significantly lower average resprout length in the treated stumps than in the control stumps, which is exactly what we were hoping to find. Looking at the other response variable, which again was number of resprouts, we can see from the graph here that the treated Norway maple stumps had a slightly higher average number of resprouts, whereas the treated winged euonymus stumps had a slightly lower average number of resprouts. But looking at the data analysis, we can see here that neither of these two differences were found to be significant. So the significant reductions in resprout length for the Manitoba maples, winged euonymus, and invasive honeysuckles are quite promising. These findings are in line with previous literature, which has also found significant reductions in resprout length for several other hardwood species. Additionally, Chondrosterium purpureum treatment was found to reduce resprout length in Manitoba maples specifically in past literature, and so this further corroborates the results that we have found here. So these findings are great news because Chondrosterium purpureum, Lalcide Chondro is potentially a great tool for invasive plant management in light of the negative impacts of chemical herbicides, and because many municipalities, conservation authorities, and agricultural landowners have expressed a desire in transitioning away from chemical herbicide use. The Manitoba maple specifically was one of the target species at Somerville Nurseries, where again, it was very abundant. So this is great news for agricultural landowners who don't want to use chemical herbicides on their land. Similarly, the winged euonymus and invasive honeysuckles were found in Rattray Marsh and High Park, making this great news for the City of Toronto and the Credit Valley Conservation Authority as well, both of whom have expressed a similar desire and partnered with us in this study in the hopes of finding significant results. The non-significant results lead us to the conclusion that Lalcide Chondro treatment is not effective in reducing the resprout length of any of the other target species, nor was it effective in reducing the number of resprouts in Norway maple or winged euonymus, at least within the time frame of this study. And this brings up an important point. Most previous studies conducted on Chondrosterium purpureum treatment looked at treatment impacts for at least a year, if not two, after the treatment was conducted. And most of these studies also found that it took longer than the 3-month time period looked at in this investigation to actually see the impacts of silverleaf disease. So it's important to consider these results within this context, as the short time frame of this study is definitely a limitation. Other potential limitations include inaccuracy in treatment application, as well as potential environmental conditions such as excess sunlight and low moisture, since we are working with a live fungus here, and both of these factors have been found to be limitations in previous studies. However, at the same time, it is of course also entirely possible that Lalcide Chondro treatment is just not effective for these species at all. Past studies have found that Chondrosterium purpureum treatment is simply ineffective for some species like seaberry, for example. And so this demonstrates the importance of running long-term studies, and it also exemplifies the importance of studying each and every target species that you want to use this treatment for, rather than just assuming efficacy based on past studies on other species. So just to summarize, Lalcide Chondro treatment did lead to significant reductions in average resprout length for the invasive honeysuckles, winged euonymus, and Manitoba maple, but did not for any of the other target species, nor did it significantly reduce the number of resprouts for winged euonymus or Norway maple. And so my recommendations for the future are to, first and foremost, continue observing these trials for at least the next 1.5 years to see if the species with significant results continue expressing this pattern over time, and to see if the species with non-significant results change over time. I would also recommend promoting the use of Lalcide Chondro to municipalities, conservation authorities, and agricultural landowners, and specifically towards those who already express a desire in transitioning away from chemical herbicide use, but also towards those who may be a little bit more hesitant to, at the very least, encourage some critical thinking about current invasive plant management strategies. In terms of future research, I would recommend investigations into the impact of treatment timing, investigations into the impact of environmental conditions, and investigations into the efficacy of Lalcide Chondro treatment for other potential invasive plant species. And that brings me to the end of my presentation, and I would just like to quickly thank everyone who was involved throughout this entire process. I would like to especially thank Rhoda, who was a fantastic internship supervisor, Allison, who was my external supervisor and has called in here today, as well as Patrick for all his helpful insights and feedback, which was essential to the formation of this presentation and my capstone in general. And finally, I would of course like to thank my MFC cohort. You have all been fantastic, and I wish you all the best in your future endeavors. Thank you.
Thanks so much, Arun. Allison, are you able to ask a question? I know you're calling in and you might have been unable to see the presentation slides. Just let us know if you can kind of chime in. If not, then yeah, or you can email Laura a question. So while we're waiting for Allison, I'm going to pass the mic to Patrick to ask Arun a question.
Thanks so much, Arun. That was great. Nice to see it all come together. Yeah, one question that came into my mind during your presentation that I don't think we discussed previously was, well, is there any risk of silverleaf disease spreading to native species following the application on the cut invasives?
Yeah, that's a great question. So obviously with any sort of biological control agent, that is of course a risk that you do need to consider. But as I mentioned, Chondrostereum purpureum is a fungus that is native to Ontario, including here in Southern Ontario. And so native species are already exposed to this fungus. More so than that, the fungus does not really spread very far. And more importantly, you need to have exposed sapwood for the fungus to actually infect the plant. So studies have been conducted on this previously, and they haven't found that this treatment really impacts any non-target plants. And just on top of that, to kind of add on to that question, it doesn't really have any toxicity in humans or animals, does not really impact any other animals either. So that is what the research has currently found.
That's great. That's helpful. I mean, otherwise, how common is this fungal pathogen? What's its prevalence on the landscape?
I don't know like numbers exactly. It is not incredibly prevalent. It is present, but it is not necessarily incredibly widespread.
John.
Is there a carrier? There must be for the fungal spore application. And I'm wondering what that carrier is and whether you really need another control. Because if you had sort of untreated and then the fungal treated, you're also adding whatever the carrier is. And that brings up a kind of related thing. There's a kind of relatively well-established practice of using fungal spores in chainsaw oil to have kind of a easy way to treat all the stumps that are produced. So I wonder your thoughts on that.
So if I'm understanding your question correctly, you're asking like what the carrier of the fungus is within the formulation of the product. That is something I unfortunately do not know. But yeah, I mean, there could be definitely other applications for it. And like, I'm sure if whatever carrier that is should be tested in future studies. But again, unfortunately, that is something I'm just not aware of.
Okay, any other?
Can we put this on there? Are you asking? That is a question you would have to ask Lalcide.
Okay, Aileen has a question. One sec, let me just give her the mic.
I'm wondering if you know about how long it takes to produce the like a batch of this, because it seems like there would be a lot of demand, and I'm
Yeah, I mean, how long it takes, I'm not really sure, but there is currently a fairly high demand for it. It is sold to a bunch of municipalities and conservation authorities. And of course, they are trying to increase their demand for it. But again, I'm not really sure exactly how long it takes to make.
Thank you. And I think Allison has a question. I'm gonna allow Laura to read it out.
Okay, Allison says, was going to ask about toxicity, which Arun just answered. Question, can you explain why the treatment timing window is late June, early July?
Well, based on past studies, early summer has been found to be the most effective time for using this treatment. I do know Lalcide is trying different treatment application timings, specifically with buckthorn. Hopefully, you know, the results show that throughout the summer it is effective. Maybe it does, maybe it doesn't, but of course they're hopeful that it does. But past literature on most other species has shown that early summer is the best treatment application time.
Great. Thanks so much, Arun. Great.
Thank you.